Welcome to Module 3. This time I split up some of the tools so that the videos overall would be shorter, so that you can pick and choose when you want to get it done. So first thing to realize is that Module 3, I'm already in my notebook, is out of order in the training manual, and that's on purpose because you're going to use some similar tools to what we just finished with in Module 2. So in Module 2, we talked about sequence-based similarity. That's really helpful information, and we compared it against like 10 other closest match bacteria based on the E-value, trying to figure out what does our protein do. And with KSED00010, we started figuring out that it's involved with chromosomal replication, and that it's this thing called an initiator protein. And we found that it shared that function with a lot of different things. We recorded the scores and the hits and the E-values and documented those. We also looked at the conserved domains, sections of the protein that seem to be conserved sequences amongst our 10 or 11 different bacteria. And we found some different things here with COGs, conserved orthologous groups, which shows that maybe they came from a common ancestor. We recorded that stuff. We did a tea-coffee alignment with those 11 or so bacteria, including our Archidococcus one. We included the multiple sequence alignment and a web logo, which all was about sequence-based similarity. The problem with sequence-based similarity is that there are sections of proteins, domains, or even just smaller sections that can be similar but end up functioning differently. So the Module 3 in your training manual is going to look at that. So we're going to be skipping in your notebook cellular localization data, alternate open reading frame, and we're going to go to structure-based evidence. And click it open, and the first tool we're going to use, and that's what this video is going to be about, is TIGRFAM. TIGRFAM is going to use something called an HMM, a hidden Markov model, to score proteins based on sections of the protein, and it's going to align it against a profile HMM in a computer or in the software and compare your sequence to it. So the software will predict for you if you have a match, specifically a functional match, meaning now we're starting to pinpoint maybe what your protein does based on its structure. So that's why it's protein structure-based evidence. So as far as how to do a TIGRFAM, the link is in your notebook. Remember, if you control-click, it will open up a new tab. And it looks like this. J. Craig Venter is a pretty famous scientist who was running kind of some human genome project stuff and has been very innovative in molecular bio, which is kind of the whole topic of this. The first thing that you'll need to know, page 59 in your training manual has what the labels are going to be that get pumped out of TIGRFAM. There are these things called equivalogs, subfamilies, superfamilies, and domains. And it's like classification in bio. Equivalogs are really, really specific in small groups. Domains are bigger groups. We'll talk about that after you get some results. So on the TIGRFAM's page, I need to, in the database, I only want to see TIGRFAMs. We're going to do PFAMs in a minute. I'm going to leave the scope set to global, and I want to change the E-value cutoff level to 0.01. If I see a lot of conservation, I can change it to our critical 0.001. Remember, the E-value is just looking at what are the chances that it's just random chance. So if you have a really low E-value, that means it's not a random chance. It's probably due to a very similar structure doing a very specific thing. So what I need from our notebook, you can see our protein sequence with FASTA header. So that is back. Good thing we documented this, right? Back at the beginning, basic info. You've been here before, not the DNA sequence. This is the amino acid sequence with FASTA header. Copy, don't include anything extra. Go back to TIGRFAM and paste it in. Everything looks nice. I'm not going to mess with it. And then it says start HMM search. You can see this is not a pretty page, but it's got information. So just basic stuff. Yep, it's KSED00010. And then there's stuff with scores, and it says there's 2 Tiger hits. And I'm seeing familiar things about KSED00010. But there's this thing, a regulatory inactivator. I haven't seen that before. So when I look over here, score-wise, There's these numbers, and then these e-values are extremely low, which means not likely that this is random chance that this matched up to, or my protein matched up to these tiger hits. Pretty well documented here. So I'm going to record those in my notebook. I've got to go back to structure-based evidence. Sorry if you're getting seasick. And I have it open. So the first TIGRFAM number is Tiger 0362. Copy, paste, save. And I love this module because it's just baby steps. TIGRFAM name. Well, this is cut off a little bit. So there's a way to deal with that. I do want the whole name. To find the whole name, you need to enter the link that is shown on the top of page 63 in your notebook. You'll see me type it in, so you can use it from here if you want, but so I'll leave it there for a second so you can use it. Pause it if you need to, but I'm gonna go there. And what I'm gonna need, this lists all the TIGRFAMs that are in there, all the tigers. So remember, I need Tiger 00362. So I scroll down. They're in order. So I'm getting close here. I'm in the 200s. 00362. DNA-A. Here's my full title. Chromosomal Replication Initiator Protein DNA-A. Okay, so this actually matches up. This is a good thing. If you're finding different things in these different tools, somewhere along the way you should be getting a really, really high e-value score, meaning that maybe it isn't based on evolution or natural selection. It might be more based on chance. So save it. And the score is back on this page. High score in this sense means good. And the e-value we're very familiar with had better be less than 0.001. And it sure is. It is almost 0. Save that. Okay, I am going to record, because there's another one, I'm going to go back and I'm going to record this as hit number 1, and I will record hit number 2 doing the same thing. The number, save it. Hit number 2. This has the whole title. I'll take the whole thing actually. Save it. And even for hit 2, this is pretty low E score, which is what we want. So at this point we have all the tiger information done. Pretty easy, right? But we also have this stuff. So when I'm back in this library where I was, if you click on this tiger, it says it's an equivalog. You can also see there's like subfamilies and domains. And in your training notebook, on page 59, it tells you what those terms mean. So an equivalog is a very specific functional protein. Okay, so it might be an enzyme, and in this case, it's an initiator protein. So that's an equivalog is the smallest group you can be in. A subfamily has a more generic set of functions for the members of the subfamily. A superfamily, which you can see down here, is a really big group with a little diversity of function, and a domain is the biggest group, which has quite a variety of functions, but they're very similar. So we've discovered something here that we want to make sure that we highlight, and it is that if I click on this tiger 00362, it gives me some more information. It is an equivalog, so it's in this really little group, and if I read, it's an initiator protein, and if you scroll down, it actually gives sources of who's studied this before and entered this in, but it gives you information about what it actually does. And now we're getting into like real, what's my protein do? DNA-A is involved in DNA biosynthesis, initiation of chromosome replication, can also be transcription regulator. C-terminal of the family hits the PFAM bacterial. Okay, so PFAM we're doing next. So now you're starting to figure out what your protein does, and when you run your Clostridium gene through TIGRFAM, you have a great chance of finding out that maybe that gene has sections that have specific and known roles. So good luck with TIGRFAM.